Assalamualaikum and hello everyone. Welcome to this video where our group will be discussing about TRP Operon. But first, let's get to know the member of this group. This group member consists of me, Sharifana Jasmine, Nur Fakira, Nurul Anis Fahana, Siti Raihan Amira, and Nomai Sarah. What is the TRP operon? It's a set of gene in E. coli that control the production of tryptophan, where it's an essential amino acid. Why is it important? It helps bacteria save the energy by turning off the tryptophan production when it's not needed. The operon operates by repression and attenuation. Repression is when tryptophan binds to a repressor protein that could stop gene activity, where attenuation is a fine-tuned regulation based on tryptophan level in the cell. So, the purpose of the operon is to ensure efficient tryptophan synthesis only when it's required.
Now, I'll be explaining the structure of the TRP operon. And generally, we all know that TRP operon consists of the several components such as structured gene, an operon model, an operator, and a repressor gene called TRPR. So this organized structure helps to allow bacteria to balance the production of tryptophan according to their needs, which we will discover their functions one by one. So the first is the structured genes. They have 5 distinctive proteins, which is TRPE, TRPD, TRPC, TRP, and TRPE. So these genes includes the enzymes that work in the sequence to produce the tryptophan biosynthesis, which means that without these enzymes, they cannot produce the tryptophan. And next is the promoter. Promoter is the specific DNA region where RNA polymerase will bind to initiate the transcription of these structured genes. So it is essentially the starting point for the gene expressions. The next, an operator. Operator plays a role for the repressor binding site so that the genes can be transcribed as a single mRNA. So not only that, promoter can act as a switch. When this switch is off, the transcription is blocked, and that will preventing the unnecessary tryptophan productions. Finally, a repressor gene called TRPR, which is located at the upstream of the operon. This gene produces a repressor protein which binds to the operator when the tryptophan levels are high, which it lead to inhibitions of the operon's activity. So, additionally, the TRP operon includes a leader sequence and an attenuator, which provide the secondary level of control. These elements, since, are regulated through the repressions, which will be explained on the next slide.
Next, I will present to you about the regulation of TRP operon. The operon is controlled by 2 mechanisms. First is the repression feedback. Second is attenuation feedback, which is the negative regulatory mechanism in the TRP operon to regulate the transcription of tryptophan, the type of amino acids in the bacteria like E. coli. So guys, what is repression? Repression is the transcription initiation in response to the intracellular concentration of tryptophan. So there are 2 things happening during repression. First is during the presence of tryptophan. Second is during the absence of tryptophan in the cell. Firstly, when the concentration of the tryptophan is high in the cell, that means the cell doesn't need tryptophan in the cell anymore. Thus, the tryptophan that present in the cell will bind to the TRP repressor. Then the TRP repressor will become activate and bind to the operator region. Thus, the operator region will be blocked. Then the RNA polymerase cannot continue the transcriptions of mRNA to synthesize more production of enzyme. So there is no production of enzyme during this case, and there are no production of the tryptophan. Vice versa, when the concentration of tryptophan is low in the cell, that means the cell needs more tryptophan. Also, during this time, the TRP repressor is inactive because there are no tryptophan bind to it. Then the operator is free from TRP repressor and RNA polymerase can continue transcription of mRNA to produce more enzyme. Thus, the production of enzyme is increased during this time to increase the production of tryptophan.
Ever wonder how cells know when to stop or start making something they need? Let's explore the brilliant system of attenuation in the TRP operon. Imagine the cell as a busy factory and tryptophan as the product. When there's plenty of tryptophan, the factory slows down to save energy. But how does it do that? Enter attenuation, a clever regulatory system. Here's how it works. The lead sequence called GRPL acts like a traffic controller. It has 4 regions, 1 to 4, that can fall into different shapes or hairpins. When tryptophan levels are high, the ribosome zips through the leader sequence like a car on highway. This creates a 3-4 hairpin which acts as a stop sign and halts transcription. But when tryptophan is low, the ribosome stalls, like a car running out of fuel. This gives time for a 2-3 hairpin to form which stop the 3-4 hairpin from appearing. The transcription process then gets the green light to continue. Now, here's the cool part. This is possible because in prokaryotes, transcription and translation happen at the same time. It's like the ribosome and RNA polymerase are working together in real time to make decisions. So, why is this important? It's all about efficiency. The cell doesn't waste energy making unnecessary proteins. When tryptophan is abundant, the system shuts down production. When it's scarce, production ramps up. Pretty amazing, right? The TRP operon is like a perfectly tuned machine adapting to what the cell needs. And that's how bacteria manage their resource like pros.
Now, I will discuss on the biological significance of the TRP operon. Firstly, the TRP operons demonstrate energy conservation. In bacteria like E. coli, producing enzyme to synthesize tryptophan when it's already available in the environment would be so wasteful. By shutting down the operons when tryptophan is abundant, bacteria actually can save energy and resources, ensuring efficient survival. Next, it also highlights the effectiveness of dual regulation. The combination of repression and attenuation ensure precise control, preventing overproduction while still responding to immediate needs. This fine-tuning is a hallmark of bacterial adaptability. Additionally, the TRP operon also illustrates how bacteria adapt to their environment. By regulating tryptophan's biosynthesis based on availability, it shows that how bacteria survive in nutrient-limited conditions, which give them a competitive edge. Beyond the bacterial survival, the TRP operon also serves as a model system for understanding gene regulation. Studying this operon can help scientists to uncover broader principles of transcriptional control, feedback loops, and energy efficiency, which are applicable to other organisms, including humans. Last but not least is the operons also inside have a practical application in biotechnology. Operon systems like this are used in genetic engineering to control protein production in industrial and pharmaceutical processes. In summary, TRP operon is not just about regulating tryptophans, but it's also about the efficient survival, adaptation, and how gene regulation works across the life.
Finally, here are the sources and references that will provide the information for this project.